Thanks for joining us today. My name is Max, and I'm part of the team here at Columbia Land Trust. We're so glad to have you join us for our webinar today, Resilient Estuaries, How Monitoring Science Improves Restoration and Conservation. And this is part of our field report series of webinars here at the Land Trust. So I'm joined here today by some very knowledgeable members of our stewardship and conservation teams. We have Monitoring Program Manager Amy Borde, Land Steward Helen Gavrilov, and GIS Specialist Elizabeth Martin. And this group is looking forward to sharing some of the learnings, photos, video footage of wetland restoration projects that have been happening in the lower Columbia River area. And in addition to that, how GIS technology and restoration ecology inform our management of the vital wetland habitats that Columbia Land Trust cares for. So this webinar is being recorded and it will be available on our YouTube channel if you'd like to rewatch it later on or share it with others who might find it interesting. We'll email you that link once it's ready. Throughout the whole session, you're welcome to submit any questions that you have using the Q&A module at the bottom of your Zoom screen. And after the presentation, we'll make some time to go over some of them live. So we'd like to extend a really big thank you to our sponsors who helped make this webinar series possible. And this year, our lead sponsors are First Pacific Financial, the Confederated Tribes of Grand Ronde, Becker Capital, and Vista Capital. Our supporting sponsors are Columbia Bank, PR Worth, Schwabe, Williamson and Wyatt, Killian Pacific, Interfluve, Northwest Natural, and Trillium Asset Management. So thank you to all of our sponsors again for making this series possible. Okay, and then before we turn it over to the team, I'd just like to share a very quick intro to Columbia Land Trust for those who may be newer to us. So for over 30 years, Columbia Land Trust has worked to protect and care for the lands and waters of the Northwest. We work in both Oregon and Washington, guided by 230 miles of the Columbia River and many of its key tributaries. Our team is comprised of biologists, ecologists, and many other experts in their fields who care deeply about the nature of our region, from coastal wetlands and estuaries to backyard habitats here in the Portland, Vancouver metro area to oak pine woodlands of the East Cascades. There are few places in the world that can sustain the diversity of ecosystems, plants, and wildlife that exist here. Together with you, our supporters and partners, we work to conserve and strengthen these lands, ensuring that our deep connection to nature thrives for generations to come. Our approach is rooted in collaboration. We partner with tribes, farmers, foresters, and local communities to make lasting shared outcomes. So, with all of that said, I'm excited to pass it off to the team so they can share a little bit more about our work today. So I'll pass it on to you all.
Thank you, Max. So my name is Amy Borde. As Max mentioned, I'm the monitoring program manager here at Columbia Land Trust. I've been here for about 2.5 years, but I've had the pleasure of working in the Columbia River Estuary for about 20 years, learning and doing research and monitoring throughout that time. So I'm very excited to talk to you about some of that learning and how we've been applying that to our work at the Land Trust. So I'm going to give you a little bit of an overview today of tidal wetlands and restoration in the estuary and why we monitor them, what we learn from them. And then Elizabeth and Helen are going to go over some more details about those monitoring metrics and methods, talking about some of the work we do with drones and other field measurements. And then I'm going to come back and talk about putting it all together and how we use some of that information, leaving some time at the end for questions. Okay, so the Columbia Estuary is located at the very bottom of a very large watershed. The Columbia River is the fourth largest river in North America. This very large system also has a very extensive federal hydropower system. So there are dams located throughout the Columbia Basin. And due to loss of salmon populations, in part, perhaps because of these dams, there's mitigation actions that are required. And one of those actions was to focus on the estuary and to find ways to improve habitat conditions in the estuary, as research has shown that the estuary is very important rearing and habitat area for juvenile salmon before they enter the ocean and begin their long migration. So zooming in now down to the estuary, there's what we consider to be the estuary is the lower 145 miles of the Columbia River, extending from the mouth all the way up to Bonneville Dam. That the river is tidal for that entire distance. And that's in part because this is a very low gradient river down at the lower part. This map was from a paper that was published in 2022, documenting many of the restoration projects that have been done throughout the estuary over the last 21 years or so. So the paper was in 2022, didn't quite include all of the projects to date, but there are now 90 restoration projects covering a little over 9,000 acres. And the Columbia Land Trust is one of 5 project sponsors that is doing that restoration work throughout the estuary. So that's just a little bit of background about how we've come to be doing some of the restoration work. We're part of the Columbia Estuary Ecosystem Restoration Program, working with a lot of other partners on this. And the reason that tidal wetland restoration is needed is because there really has been a dramatic loss of this kind of habitat over the last 150 years or so. About 68% of what used to be tidal wetlands are gone due to various development and diking and farming and reasons such as that. So a loss of over 50,000 acres. So this restoration program is very important to try to restore at least a portion of that to regain that really important habitat. So over the years, myself and other colleagues in the estuary have done a lot of research to try to understand this very dynamic system. As I mentioned, the tide extends all the way to Bonneville Dam, but it's also a very big river. So we have that interaction between the ocean tides and the river flows that really creates a very strong gradient of change as you move in terms of hydrology, as you move from the mouth up to the dam. So we've really done a lot of work to try to understand how that hydrology affects the plant communities and the wetland communities within this system. So we've looked at a lot of what we term reference sites, sites that have been kind of least disturbed, to really try to understand that dynamic and those processes. So this is a diagram that I made. This is work that we've done looking at those reference sites over the years and really trying to get a better understanding of how the hydrology and these what we call datums, or mean average water levels, correspond to changes in wetland plant communities. So as we move up the elevation gradient from low marsh to high marsh to shrub wetland, up into forested wetlands like such as Sitka spruce swamps, that's all based on how the water level interacts with the different elevations, and those are the plant communities then that establish within those different zones. So this is important to really help inform restoration. We want to understand what plant communities we can expect, or actually even create our designs to ensure that we're going to get the plant communities that we are targeting. So Columbia Land Trust has just over 4,700 acres and over 26 properties that are in this tidal floodplain of the Columbia Estuary. Uh, some of those have been conserved and are relatively intact. Most of those are areas of Sitka spruce swamp, which is really kind of just a remnant habitat. There's not a lot of that habitat left in the estuary, so we've been able to conserve some portions of that. And then we have about almost 1,400 acres that we have restored already. Another 1,000 or so are in the design phase, and then we have 1,000 or so acres that we're looking at for potentially restoring in the future. So I'm going to zoom in now to a little bit closer to one of our sites. This is Carey Island. It's in Westport Slough. It's located about river mile 40 or so along the river, right near Puget Island, if you're familiar with that area. And this is a aerial photo of what the site looked like prior to restoration. It was essentially a cow pasture with some areas of forested areas. And then post-restoration, this is what it looks like now. So these are excavated channels that you can see in this drone imagery. And we also use some of that excavated material to create some mounds that are now planted with willows. So this is really now a really beautiful matrix of habitats at the site, from low marsh elevations all the way up into shrub and forested wetlands. I'm going to give you an even closer view on the ground. This is a native wapato, which is a very important native plant. It's also been harvested over the millennia as a first food by the indigenous tribes. It creates a tuber, which is like a kind of starchy tuber, like a potato. It was an important food source. And just to give you a little taste of what these systems are like, I took this video walking through the wetland. The wapato is nearly head high for me, about 5.5 feet tall. And they're very muddy, very wet systems, very hard to walk through, but really beautiful and really very, very productive and full of life. Okay, so why do we monitor? One reason is to inform future restoration. We want to know, how the things that we've done at restoration sites are evolving and whether they are doing what we expect. And also, we can learn from these sites by looking at the elevation and the hydrology and the plants to inform what really dial in those things that we need to do at future restoration sites. Okay, and also along with that is evaluating the effectiveness of the restoration that we have done to see how it's moving along the restoration trajectory as these systems change over time, and also to address uncertainties. So these are things like climate change and how these systems may respond to climate. Also things like invasive species and what the effects might be of different invasive species and how we might learn to control them, which I'll talk about a little bit later. All right, so this is just wanted to show some pictures that we've taken at the site. We monitor a host of metrics, which Elizabeth and Helen are going to talk about next. But really, observations are also a type of monitoring. And these are just examples of some of the things that we have observed when we're out there. And it's something that really anybody can do. And just documenting those things that are observed are very, very important. So we see a whole host of species, or we see signs of them. The upper right is the cougar tracks that I very alarmingly came across right as the tide was going out. I knew it was very fresh because the tide had covered that area very recently. We see beaver signs all the time. We see juvenile salmon, a whole host of species. So we focus on monitoring the habitat because we can then rely on other studies and literature that have linked these types of habitats with fish and wildlife populations. Particularly, this is true for salmon. Salmon are very hard to monitor. It takes a big effort. So there's been a lot of expense and time put forth in the estuary to monitor those fish populations and try to link that to these certain types and elevations and habitats along that estuary gradient. And we've been fortunate to learn from that. And when possible, we do work with research collaborators to answer specific questions and really take as much as we can take opportunities to incorporate experimentation into our restoration design and learn and try to address some of those uncertainties when possible. Okay, I'm going to turn it over now to Elizabeth.
Thank you, Amy. Hi, everyone. My name is Elizabeth Martin. I'm the GIS specialist at Columbia Land Trust, and today I will be talking about one monitoring tool that we've been developing that is drone imagery classification. I'll be going over the basics of what image classification is, the work that goes into performing image classification, and what we hope to use it for. So you can go next slide. Image classification is the process of training a computer to identify and map groups of similar objects from aerial imagery, typically from satellites or drones. This photo on the right is an example of before and after image classification. So you can see it's drawing features like trees, roads, and buildings based off of the imagery. Now, this is a useful tool because it can map much larger areas than if someone were to do this by hand. And it can also pick up on patterns not obvious to the human eye thanks to the addition of multispectral imagery. And the uses of image classification are pretty widespread. It can track land use change, help with managing infrastructure, measure the impacts of natural disasters, or more relevant to our work at the Land Trust, it can help with mapping plant communities at a landscape scale. Next slide. So how can we use image classification to support our work in the estuary? So as Amy's touched on, the picture on the right is one of the sites we're doing the image classification at. It's part of the 4,700 acres of tidal wetland we manage. And these sites ideally are inundated during high tide, crisscrossed with channels, and heavily vegetated. These features make for great salmon habitat and other benefits like flood mitigation and water quality, but it also makes it incredibly difficult to walk around. It looks nice from the air, but Amy showed us the first person view, and this is my view of Amy in the wetlands. So you can see tall vegetation, water up to your knees, really hard moving around. So the main goal of this classification program is to be able to map plant communities across entire sites at places like this where walking the whole site and kind of getting your eyes on the whole area isn't feasible. We hope to create a more complete land cover data set than we would get from traditional monitoring techniques that are really good about giving you a lot of detail in a small area. This is more general detail over the entire site. And then we can use that data to help us better understand our sites, inform management decisions, monitor changes over time, and especially following restoration work. Next slide. So to do image classification, first you have to collect all the data. A large part of the field data collection is gathering vegetation points. That's the picture in the middle there of Amy getting vegetation points. These are point locations of different plant species taken all across the site. We focus on the species we're most interested in mapping, and we take as many points as we can because these are used to teach the classifier what to map. We also collect our drone images. So image classification can be done with satellite imagery, but we use a drone because we can get a lot higher resolution, and we pick the time of year we can fly to be best lined up with different plant phases. The drone flies a grid pattern over the site and takes overlapping photos at regular intervals. Typically, it ends up with about 1,000 photos for a 100-acre site. These photos are captured in red, green, and blue wavelengths, which is your standard true color that we all see. But they also capture near infrared and red edge bands, which are shorter wavelengths not visible to the human eye, but they reflect off plants in a way that can help differentiate between species. Next slide. So once we collect all the data, typically it takes 4 or 5 of us about a day in the field to get all the data we need. Then I head back into the office to do all the processing of the data. The first step is taking those 1,000 or so photos and stitching them into one cohesive image that we call an orthomosaic. The image on the left here shows what that process looks like. Each red dot represents a single photo, and the end result is one single image. And in that image, each pixel is about 3 centimeters in size, so we're picking up a lot of detail. And each pixel has 5 values representing those 5 wavelengths that the photos are taken in. Then we create what we call training polygons using the vegetation points we took in the field. These polygons are examples of each plant community or species we're trying to map. So for example, Amy showed us that picture of Wapato. We want to be able to identify it with the classifier. So while we're in the field, we took a lot of points of Wapato that we come across. Then we take those points, overlay them on the image we created when we stitched our photos together, and we outline areas of Wapato using the points as reference for what Wapato looks like. Then you kind of do it for every other plant species. You can see how it adds up into a lot of work. Next slide. So something great about having multispectral imagery, those near infrared and red edge bands, is that you can use different combinations of the bands to create additional indices that reveal patterns not apparent in a single band. These can indicate factors like chlorophyll content, color variation, or canopy height, which gives the classifier more information to work with when differentiating between species. So I will calculate an additional 5 or so indices based off the first 5 bands we got, and then I stack them all together into one image. So now we still have one image, but each of those pixels has 10 values associated with it, each representing a different wavelength or indice that we're interested in. Next slide. So everything up until this point has been data collection and processing. We haven't actually done any image classification, but this is where the fun starts. We can actually run the classifier. It's all happening behind the scenes in a big mess of code, but I'll walk you through kind of what it does. So the first step is it takes those training polygons we drew and overlays it on the full image and extracts the values of the pixels that overlap with each of the training polygons. And this builds a list of all the different pixel values that correspond with each plant species. You then take that list and feed it into a classifier, which is essentially an algorithm that looks at the values stacked in each pixel for each plant species, and it identifies which pixel values are representative of that species. And then through this process, it builds a dictionary of defining values for each species, and this is called training the classifier. Then you take that trained classifier and go back to your original big image and feed it through the classifier. And what it does is it goes pixel by pixel over the full image. It looks at each of the values within the pixel in that image and compares it to the dictionary it built in that previous step. And it predicts what plant species it thinks that pixel is based on its values and what it learned in the training step. It then draws a new image that's a copy of the original, except that each pixel has the added value of what plant species the classifier thinks that it is. And this is something that sounds simple, but depending on the inputs you put in and the parameters you set, you can get really different results. So we end up repeating this over and over again until we get just the right combination of inputs that it gives us a good looking image. You can go to the next slide. So the result of this is the same image you started with, but now instead of it being in true color, you get the plant community of each pixel drawn for you. So this is an example of the results. On the left is your true color image. It kind of just looks like a whole bunch of blobs of green. You can see the river, but that's about it. On the right, you can see it's picking up on different patterns and identifying individual species within that mix. And it looks like you can see the river running through and everything. So once you have a site that's fully classified, you can do things like calculate how many acres of native plant coverage there is, or identify patches of invasive species before they become a bigger problem. Or you can document the impacts of restoration on the plant communities, or like Amy was showing the different elevations in the different plant communities. She can use it to compare it to a topographical data set and see if the site is following the trends that we think it will. And especially if you repeat this every year, every few years, you can get a really comprehensive data set of what your land looks like and how it changes over time. So long term, this is something we're hoping to incorporate regularly into our monitoring program and have a really solid record of what our sites look like. So I will now hand it off to Helen to talk about some of the other monitoring techniques we use. Thank you.
Thanks, Elizabeth. Hi, everyone. It's good to be here today. So Elizabeth shared about a pretty exciting cutting edge monitoring method that's still being developed, but I'll talk a bit more about how we've been doing our long-term monitoring on the ground. Next slide, Amy. So we look at a variety of metrics to get a sense of how the landscape is responding to our restoration efforts. Our tools and methods range from very basic items found at hardware stores to very high-tech instruments. And primarily, we look at sediment accretion, or the buildup of sediment in the wetlands, and we look at changes in channel morphology, the shape, the depth, and the width of the channels, and we look at hydrology, the water levels and temperatures. And that's what I'll focus on today. But we also keep track of elevation, and we take photos at specific points on the site to compare how things change visually over time. And we conduct vegetation surveys to document how the plant community changes. Next slide. So we measure sediment accretion rates across our wetland restoration sites using good old-fashioned rulers, PVC stakes, and a construction level. And we install several transects across each site that are 2 PVC stakes about 1 meter apart and driven about 1 meter into the ground. And each monitoring visit, we put a level on top of the stakes and we measure the distance from the level down to the ground at every 10 centimeters. And these distances inform us of how much sediment has accreted over the past monitoring period. And over time, we can calculate the rate of sediment accretion using these measurements. So here's an example of a plot from one of our transects at Candle in the Grays River watershed. And here you can see the timeline on the x-axis, and on the y-axis, you see the change in the distance from the level to the ground since the time of initial restoration. So from this, we can calculate the annual rate of accretion. And in this case, this transect measured an annual accretion rate of about 1.5 centimeters a year over 10 years. Measuring sediment accretion rates on our wetland restoration sites helps us learn how much sediment from the adjacent river or stream is being captured by the wetland with each tidal cycle as the stream or the river overflows. And it also helps us to compare how the wetland elevation stands up to projections of sea level rise. Next slide. We also look at how the shape of our restored channels change over time, and we do this by using a super precise GPS unit called an RTK to take elevation points along a transect that crosses the channel. And this essentially maps what we call channel cross sections and results in plots like the one you see here. So that plot is kind of like looking at the channel from the side. Like I like to think of it like looking at a slice of cake from the side. This channel cross section from one transect at Candle with the distance along the transect on the x-axis and the elevation in feet on the y-axis. So we have these PVC stakes installed on each bank of the channel, and then we try to walk in a straight line from one stake to another. And we take elevation points using the RTK whenever we see or we feel like a slight change in elevation. And this plot shows data points in the different colors from different years that we've collected the data. So the gray line is the oldest, right after restoration, and the yellow is the most recent. So you can see that the shape of this channel has changed pretty dramatically over time. And you can also see from this plot that the elevation of the banks has changed as well. So these plots I find the most fascinating. I love looking at them because they really kind of show, they really demonstrate kind of the change in the channels and how things just change over time. It's pretty awesome. And the smaller plot on the right shows calculated metrics that help us quantify that change over time. So those metrics are the channel width, channel area, the depth, and then the width to depth ratio. So measuring channel cross sections helps us visualize the changes in the shape of the channels. It helps us assess elevation changes in the floodplain and compare channels to each other across the site. And this ultimately helps our engineers know if we've achieved our goals in the restoration design, and it helps them inform their future restoration design. Next slide, please. When we monitor hydrology on our restored wetlands, we look at the elevation of the surface of the water, the depth, and the temperature. And we do this by deploying several long-term sensors across the site that takes readings at specified intervals. Usually it's like 15 or 30 minutes. And then we go back periodically to download that data and process it and convert it into usable values that we can plot. In addition to those readings, we take various measurements and do some math, and it helps us to interpret the data. And the result is a plot that you see on the screen. So the top is a plot of the water surface elevation. The timeline is on the x-axis, and the water surface elevation is on the y-axis. The plot itself looks like a fun sound wave type of shape because it's a tidally influenced site, and so you can see the daily high tide and low tide cycle. The plot on the bottom depicts water temperature, and both of these plots are over a 4-month period from May through August 2023. So measuring water surface elevation and the depth and temperature helps us understand how hydrology changes throughout the restored wetland over time, and it helps us see how frequently the site is inundated and how it's affecting the plant community. So with that, I'll turn it over to Amy, and she'll talk about a couple more specific examples of how monitoring has helped inform science and management.
All right. Thank you, Helen and Elizabeth. Those were great overviews. I'm going to talk now about an example from the Grays River area. This is going to be looking at the example is one where we were able to use data that we collect, such as some of the things that Helen just talked about, to really start to address some concerns that the community has regarding high sediment loads and sedimentation in that river. So I'm going to talk just briefly about how water moves sediment. You might recognize this is the same plot I showed earlier about how water affects plant communities. Well, it also really dictates the accretion rates that we see in these areas. So the areas that are inundated very frequently at the lowest elevations tend to have the highest sediment accretion because of the frequency that they are receiving the water and the sediment that comes along with it. And as we move up the elevation gradient, we typically see that there is it's inundated less frequently and less sediment is deposited in those areas. So sediment accretion is an important process for wetlands to be self-maintaining over time. And gradually, they gain elevation and succession occurs from one community to the next. So over a long period of time, sites will gradually shift from, say, a low marsh community to a high marsh community as that elevation increases. And that accretion rate then slows down too. So once a site reaches its climax wetland community of forested wetland, it often stays at that community for a very, very long time because accretion rates are much, much slower. So Helen mentioned the sediment accretion stakes that we use in our work. And I just wanted to mention that that method has been evaluated by some of our colleagues in the estuary and compared to a more robust system that is shown there on the photo on the right. It's called a sediment elevation table, and instead of driving PVC into the ground for about a meter, it's a steel rod that is driven into the ground, sometimes up to as much as maybe 7 meters to the point of refusal. And it's capped with concrete, and a receiver is placed on it so that it's a very stable benchmark almost in the wetland. And then a receiver is set on top of that that has these 9 pins that are moved at very specific locations and drop down in a very measured and exact way. And what they found after looking at a comparison between the two methods at multiple sites over multiple years is that really the results were very comparable. So this was a great indication that this was a good method to be using, much cheaper, much more quick and easy to measure for all of the practitioners that are working in the estuary. So you may recognize this as the candle site that Helen talked about. I'm showing specifically the accretion stakes that we have at that site. There are 9 of them, and they span the elevation gradient at the site. And again, as Helen showed, this is from one of those. So I've shown here that rate at that particular one is 0.7 inches per year, which equates to about 8 inches over that 11-year period. So if we take that data then and look at it, so each dot here represents the annual accretion rate for that set of accretion stakes. And because they occur across the elevation gradient, that's what I'm showing here in the x-axis, you can see that there is in fact that relationship that I described. As we move up in elevation, the accretion rate decreases. So if we take the average of, this is a just kind of quick and dirty way to look at how much accretion is happening over the entire site. So I took the average of those accretion rates, and which is about half an inch of sediment per year. I multiplied it times the area of 140 acres of the site and came up with a really large number and converted that to dump truck loads. And it's about 940 dump truck loads of sediment per year that is accumulated within just this one site in this particular estuary. So this is higher than what happens in some other areas of the estuary because of the high sediment loads in the Grays River. And partly it may also be because it's a restoration site, which research has shown that because sites are diked, they subside over time, and so their elevation is lowered compared to what they were prior to being diked. And often, once restoration occurs, those accretion rates are much higher than they are in natural restoration areas, or obviously once they reach back to their original elevations of, in this case, a tidal forest. So those two things combined are causing some of these very high accretion rates that we're seeing there. But the point in this particular case, we did these calculations and talked to the community because there's concern that the riverbed is starting to fill with sediment, and Grays Bay is starting to fill with sediment, and it's potentially exacerbating flooding problems. And so we just wanted to be able to tell the community how much sediment these floodplain wetlands have the capacity to hold, and how much they do in fact trap over time throughout a single year, and how much that accumulates in multiple years to help to reduce that sediment load as the water moves through these floodplain wetlands. All right, another example I'm going to talk about is a study that we did to try to determine methods to reduce reed canary grass. So why is reed canary grass a problem? Well, it's a non-native invasive species. It's found within the mid to high elevation marshes throughout about 125 miles of the tidal freshwater portion of the estuary. And of particular concern are restored areas that are very susceptible to invasion because there's been a disturbance with the restoration and there's such a large seed source in the estuary that, and it's a very strong competitor, so it very frequently um, invades restoration sites. So it also has many ecological effects that we're concerned about. Obviously, lowering plant diversity, but it can reduce channel formation. Because it forms such a dense mat, it reduces access for juvenile salmon or other fish. It also affects the food web. In the decomposition of the plants is different than native plants. It's slower, lower quality food source. And there's also fewer macroinvertebrate prey species that have been found. That was part of a study that was documented by the paper I'm showing there. So what do we do? Well, it's a challenging problem. And we conducted a very extensive literature review, and we also interviewed regional practitioners. There's not a lot of information that's out there for tidal wetlands. A lot of the literature is really focused on wetlands that are not tidal, but just in other parts of away from tidal inundation, but also in other parts of the country, in the Midwest. So we combed through all of that data. We wrote this paper to document what the findings were. And basically we found that some of the methods that are used are manipulating elevation. You can excavate the site to be lower so that lower marsh communities will be able to come in, or also increase elevation, creating mounds for woody species like shrubs and trees that can eventually shade out reed canary grass. But the problem with those two methods is that this is at the expense of this very productive high marsh system. So we really wanted to try to see if we could find a way to control reed canary grass while maintaining this high marsh community and convert it to native species rather than reed canary grass. So we decided to implement a targeted study that used herbicide treatment and integrated that with native seeding. So we chose to implement this site at two sites, the Candle site we've been talking about and the Carey Island site I showed the pictures of earlier. And we developed an experimental design with some of our colleagues at Pacific Northwest National Labs. And we decided to implement this at two different elevations, kind of the mid-marsh elevation, where reed canary grass is not quite as competitive, and the high marsh elevation where it really tends to be dominant. I'm not going to go into a lot of detail about the study itself and go right to the results that we found after 6 years of monitoring the vegetation every year during the peak of the growing season. We did implement spraying in two different treatments. One was 2 times in the spring and the fall, and then compared that to a treatment that was spraying just in the fall. At the Candle site, we implemented both. At the Carey site, we only implemented the fall treatment because it was a newly restored site, and reed canary grass really hadn't gotten established fully yet. And so we felt that it would probably respond to just that once annual treatment. And in fact, that was the case. So what you can see here in these plots, the colored blue and green are basically the treatment plots where there was spraying conducted and seeding. You can see there wasn't a huge effect of the seeding. We didn't see a lot of difference between the plots that were seeded and those that weren't. But we saw a dramatic effect of the spraying, reducing. So this is particularly showing the cover of reed canary grass in the treatment plots in the color, and the gray plots, the gray line represents the cover of reed canary grass in the control plots where we didn't do any spraying or seeding. So on the y-axis, this is cover ranging from 0% to 100%. So you can see that the cover was very high in the control plots where we didn't do any treatments, and very low in the plots where we did. And in fact, we did find that at Candle, the fall only treatment really was not as effective as the spring and fall. So where plants are really established and the population is very dense, that twice annual treatment is really what is required. We sprayed for I believe it was about 4.5 years. And then we monitored for an additional 2 years after that. So this end of the plot shows that time, even post-treatment, we really saw consistent results that persisted. The question is how long that will persist over time. So we're going to continue to monitor these sites into the future to just document and learn more from this study. So one interesting thing you might notice is that at the Cary site, at the lower elevation plots, we started to see a reduction in reed canary grass also in the control plots where we were not treated in. So we wanted to dive in a little deeper and try to understand what was going on at that site. So we looked at the hydrology. So this plot is representing inundation. So this is taking the data that Helen talked about, where we have that 30-minute or hour interval of data that over the course of a year, and we calculate how much of the time that water level is over a certain elevation. And that's the elevation is shown on the x-axis, and the frequency of inundation is shown on the right. So these lines represent the inundation frequency along the elevation gradient at the two sites. And you can see it's quite different between the Candle site and the Cary site. I'm showing here the elevations of the site, of our study areas at each site. And you can see they cover a very similar elevation range. However, the inundation that is occurring at those sites is quite different. So really, what we determined through looking at this, that we think that 50% inundation frequency is about the threshold for reed canary grass. So you can see at the Cary site that when we initiated the study, it was very close to that lower threshold at the lower end of the plot. The plots ranged in elevation from about 1.8 meters up to 2.4 meters. That's that extent that we're seeing there. And that was similar at Candle. But the inundation was much higher at Cary, coming close to that 50% threshold. And then over the course of the study, the elevation moved. I don't know if you caught that, but at some of the mid-elevation plots where we saw that change over towards the end of the study period, we went back and we measured all the elevations of our study plots. And in fact, the elevation move decreased at some of those plots because there's what we observed was a kind of small channel, a network of small channels that was developing over the site. And that reduced the elevations a little bit further and really pushed it beyond that 50% inundation threshold, where it was inundated even more frequently and giving that native population even more of a chance to be competitive. And reed canary grass just does not occur really below that threshold. So these were some really interesting findings. We're working on getting this published and hopefully the paper will be out soon. I wanted to end with just the photos of some of what we see at that site. The photos on the left are all the photos at the beginning of the study. So even at Cary, the reed canary grass wasn't really established, but it was definitely present at the site. And then these are all towards the end of the study and the last years of the study, and you can see the Wapato in the top and the bottom photos at Candle Cary, and then the Candle high elevation really shifted to native grasses. Rice cut grass is one of those, and native sedges. Um, so it was just very exciting to see. It was something we learned a lot and look forward to learning more even over time as we continue to monitor. Okay, I'm gonna stop talking now and give you all a chance to ask some questions.
Yeah, thanks so much, Amy, Helen, Elizabeth, for that really insightful presentation. It's so cool to learn more. Even, having talked to you each about it in the past, still you learn so much from what you're doing out there. And some of those examples are just so neat. Some of the research you're working on to see how it all comes together to inform the Land Trust's work is really a neat thing to see and learn about. So we definitely appreciate you sharing all that info. We do have a few questions from some of the attendees that we'd like to get through with some time we have left here. Let me start with this list here. Um, Elizabeth, I'll start with you. We have a question from somebody asking about some of the data from the image classification that's used and wondering how much of the data set you hold in reserve for testing the classifier.
Yeah, so we are just reaching the part of the process where we're having to test the results. So we don't technically have a number yet, but everything has suggested, not everything. What we are planning on doing is using 30% to test the classifier. So for those who are unfamiliar, you train the classifier, but you also want to make sure it's accurate. So you reserve some of the data, you don't use it to train, and then you compare the results from the classifier to the data you have to make sure that the classifier is being accurate. And so we're planning on reserving 30% of the data.
Awesome. Thanks, Elizabeth. Um, and then just to follow up on that is, are you able to use this technology in other landscapes, other ecosystems, other places in the Land Trust's service area?
We haven't yet. We chose the estuary partly because we've done such intensive work there, but it's also flat and has no trees, which makes it easier to navigate by drone. So the potential is there for like forestry monitoring and that sort of work. We just haven't explored it yet.
Awesome. Thanks. A question about mammals and referencing elk as well in particular, and if they're able to inhabit the estuary like other wildlife species, or if maybe the conditions are not the most ideal for them if they get stuck in some of these sites that we are stewarding.
Uh, yeah, I can give a little bit of an answer. We see elk at these sites, not real frequently, but we do see them. We see signs of them, and it's typically at the higher elevations that aren't inundated quite so frequently. Um, but they do, kind of like to bed down sometimes in some of the higher marsh areas. So we do see them. It does, the inundation and the tide does limit when they can be there.
Thanks, Amy. Let's see. So a question about flooding. And in addition to monitoring sediment accretion, whether or not we're also monitoring flooding that's happening at our sites, or if we rely on other public flood maps primarily for that type of info.
Yeah, that's a great question. Um, just a couple of nights ago, I believe there was a pretty big flood that occurred out in the Grays River and in many, many areas of Western Washington and Oregon. So what we've been doing is working with some of our research colleagues who do modeling work. And so we've been providing the data that we collect to those modelers so that they can um, create the model and then evaluate um, different scenarios, so different flood levels and different sediment accretion rates, and trying to incorporate that all into the models. We can also then look at different scenarios of restoration and how that might affect um, flood levels and if it will, in fact, reduce floods. That's been shown in lots of places throughout the country that wetlands, when floodplains are restored, they help to reduce flood levels. It's complicated in this system because of the tides and the Columbia River flow and watershed flooding that all occurs at the same time. So these models are really important to try to understand that better. And also it allows us to look at what other, possible scenarios might be able to help alleviate some of the flooding concerns.
Awesome. Thanks, Amy. So many great questions. Thank you all so much. Let's talk about maybe some sediment in the grays, and just kind of clarifying what some of the sources are for high sediment loads in the Grays River, and maybe just talking a bit more to the strategies to help reduce very high sediment loads
Yeah. Um,
and
there's
its impact on
multiple,
restoration. Yeah.
yeah, there's multiple sources. It's increased over time due to changes in land use in the watershed primarily. Um, there's, areas of intensive logging that can contribute additional sediment into the watershed, into the river. There's also, natural sources of landslides and things like that. So it's a combination of things, but often it is tied to those land use practices um, in the watershed. So uh, it's challenge. That's a challenging thing to try to address. One of the things that we are doing at the Land Trust is trying to acquire some of those lands that are in the watershed in the upper forested areas to try to maintain, some forest cover and reduce some of that uh, sediment loading that might be coming from runoff and those watershed processes during storms, particularly. Um, uh, I think that's that. I'm not sure if I fully answered that question, but restoration is another thing that we are looking at to try to improve, retention of sediment in the lower part of the river.
Thanks, Amy. Let's see what we got here. I think this another question about accretion is here as well, and whether or not um, you were expecting higher accretion rates at the lower elevations, or if you expected higher accretion at higher elevations. This particular question, the person says that they recall some of OSU, Fish and Wildlife Service, coastal wetland monitoring from a few years ago that were showing higher accretion rates at higher marsh elevations.
Hmm. Um, well, my expectation, or what we thought and had heard from some other studies was that accretion uh, decreases as you go up in elevation, and that is in fact what we saw. I know there is some variation, sometimes at the very lowest elevations where plants are rather sparse. Sometimes accretion rates are actually not that high because there's not the plant community there to trap as much sediment. So there is some variation within, because of some of those kind of things. But on whole, across that elevation gradient, all the way up into forested wetlands and into low marsh, yeah, we typically see that it decreases with increasing elevation.
Awesome. Thanks, Amy. Thanks, everybody. have a hunch it's probably the most what we're going to get to today. We had so many other great questions, so thank you all for sending them in. And if you would like to follow up and have our staff take a look at them, you're welcome to shoot us an email. We'll also be posting this webinar live after this as well, or excuse me, the recorded version of this webinar. Um, once we're all wrapped up here. So we just want to thank you all again. Thank you to our presenters for taking the time to share this great info with us all. And thank you to those who joined us. Keep your eye out for any future upcoming webinars as part of this field report webinar series. And if you're feeling inspired to learn more or get involved or support our work, we'd love to hear from you. You can visit our website, columbialandtrust.org, or stay connected with us on socials or on our email newsletter. So thanks again, everybody.